Okay, so thanks very much for the invitation. It's very privileged to visit Russia for the first time and to visit collaborators in the Metaspades team. So Ala, Anton particularly, great to see you. So what I wanted to talk to you about is the fact of the insights we've managed to gain from investigating the human gut microbiota. And I'm going to tackle this from a metagenomic and genomic perspective. So just to give you a bit of background, so I come from EMBL EBI. So this is part of the European Molecular Biology Laboratories. I'm based just outside Cambridge in the UK. And at the EBI, as Mick suggested yesterday, we have a lot of compute, but we also run a lot of services and we house a lot of data. And so my team has two roles. We're both a service and research team. And as part of the service team, I run a number of different resources, protein annotation resources like Interpro and PFAM, RNA families, RFAM and RNA Central, and also Magnify, which is EBI's metagenomics analysis platform. So Magnify I'm not going to talk about much, but just to point out, so this is a free to use service for archiving, assembly, analysis and browsing of microbiome data. We'll do amplicon metagenomic, metatranscriptomics. Assembly is a relatively new service for us. We introduced it about 18 months ago, and we've already got 14,000 assemblies up online that are annotated. There's about another 10,000 that are stuck in ENA because we try and upload those before analysing them. And this is all, virtually all of those are Metaspades. So that's just a little shout out to the Metaspades. So we've already heard from Pilar about the importance of the human gut microbiome. And we know from Pilar's presentation, it plays a very key role in early development, food and nutrition, but we also know that the human gut microbiome plays a really important role in many different diseases, things like type 2 diabetes, IBD. There's other cases of it being linked to colorectal cancer. There's this gut-brain axis with Parkinson, and we see that organisms are sort of correlated there. We don't always necessarily know the functions they're performing. I mean, one of the best examples to date of a disease that we know is things like C. diff infection, and we know that we can recover a C. diff infection with things like fecal microbiome transplants. And actually, if you look back through the literature, there are examples dating back to sort of 1958 of fecal microbiome transplants. But if you actually go back to the history books, there are cases in sort of medieval times of essentially fecal microbiome transplants happening in sort of monasteries throughout Europe. So we know it's very important, the gut microbiome. And so the question is how much do we understand? And there have been a number of papers over the last 10 years or so where there have been new ways of applying new and novel ways of culturing the gut microbiome, and papers coming out such as these two. And these have suddenly, given rise to sort of new collections. And so I work at EBI, and just across the road from me is the Sanger Institute where Trevor Lawley works. And he's worked a lot in sort of techniques of using different growth conditions. So he really expanded up the thermocutes, the sporulations. And we really we have the luxury of having conversations. And so one of the questions we sort of argue about is, so he keeps releasing these large collections in his book, so a few hundred isolate genomes at a time. But the question we really wanted to understand is, okay, how many more? So if you take an average adult individual, there's about 400 different species in their gut microbiome. We've seen multiple collections come out, but each time these collections come out, there's a new wave. But how many of those organisms are left to actually find and identify? We don't really know. We know from 16S we have an idea, but generally 16S doesn't give you enough resolving power to get down to species level, let alone strain level. So there have been 2 more papers this year. So Trevor released another collection, and that encompassed 739 genomes. The BGI also released 1,520 genomes. So does that mean we've got everything covered? I mean, numbers would stack up. Now, within those sets of genomes, there's lots of strain variants. So what we wanted to do before Trevor's paper came out, we were talking, he gave us access to these genomes. And so what we wanted to do is actually start addressing this question. And this actually also helps us, from the informatics point of view, to address a different question. So we can try and find out how much is left. So Trevor wants to retire soon, I'm sure. You know, can he sort of give up in 5 or 10 years? Who knows? But the other part is, a lot of the time, people assess metagenomics analysis, they use a mock community. And that mock community is used as a benchmark for assessing how well your methods work. What we thought we could try and do is, if we took a completely de novo approach to assembling and kept all of these genomes in our back pocket, we would hope that we would see some of those genomes again, and then we could compare how that de novo approach compared to these isolate genomes. And we really want to have this build up this collection really such that we can start going from this correlative approach of microbiome analysis to actually starting to build up really sophisticated mechanistic models. So when people start looking ex vivo to exactly what the gut microbiome is doing, they often are using just a handful of isolate genomes and using what they learn from those handful of isolates to actually infer what the gut microbiome is doing. And that always feels very uncomfortable to me. And so having this complete collection will allow us to understand a little bit more about the variation, if we see any more variation. So we weren't sure when we started out how much more we would see. So what we decided to do is to take as many public metagenomes that were from the human gut, and we skewed our collection very much to samples that were typically annotated as being from adults rather than infants. So that's an important distinction. And what we wanted to do is just take the raw reads, we'd run metaspades at scale to get the assemblies, and then we would bin those. Now this is very much like Mick talked about yesterday. Now we were doing single sample assembly and single sample binning, and we used MetaBat 2 for that. So once you've gone through that process, we want to understand how good these are. And we wanted to first of all just take a very agnostic way, not look at these genomes to understand, you know, how good are these bins at representing a genome. So there's a tool called CheckM that uses a series of marker genes to assess completeness and contamination. Titus mentioned it yesterday, for those of you who may not have been in Titus's talk. So these are single copy marker genes. So what you want for a specific clade is you want to match every single one of those marker genes once. So if you've got 10 marker genes and you match 9 of those, you'd be considered 90% complete. If you match one of those marker genes more than once, then you're considered to have contamination. So CheckM can also give you a crude taxonomy based on that marker gene, so it looks and uses clade-specific sets, and that can give you an insight to the taxonomy. So that was our first step, and this is what you get out. So on the left-hand side is completeness along the x-axis plotted against contamination. And from our 13,000 assemblies, we got 12,000 nearly where we could actually get assemblies, where we could actually get bins. Now, some of those samples were mislabeled, so some of them were amplicons, so when you try and assemble them, you don't get anything. Some of them were just a bit ropey in terms of sequencing. We just didn't get long enough contigs, so we will only bin if we've got contigs over a certain length. So we put a threshold of 10,000 as for the longest single contig, so 10,000 base pairs, and we also put a limit of 2,000 base pairs as a minimum contig that would go through to the binning. So not all data sets we could get good assemblies, but from that we got nearly 250,000 bins out. So we applied check-m to all of them, and this is the distribution on the left-hand side. Now, you'll see a huge smear of spots, and what we've done is color-code these spots according to a number of criteria. So down the bottom right-hand corner is green, and these are where the genomes or the bins are considered to be 90% complete and less than 5% contamination. This is according to the MyMag standard that's in the community. As part of that MyMag standard, there's a sort of, and that's sort of considered high quality. There are some additional things you need to look for to say they're really high quality. But there's another, the second sort of medium quality standard is to consider the blue areas and just look at both for the time being. So in that, you're having 50% complete and allowing 10% contamination. Now, within our team, we don't particularly like that. And so what we actually do is we penalise the contamination. So this is actually a metric that Donovan Parks came up with, who actually is the author of check-m. And so what you do is you multiply the contamination by 5 times, and you subtract that from the completeness. And that's what corresponds to this dark blue area there. Now, for me, that makes more sense. I'd rather accept a genome or bin that is 50% complete and has 0 contamination, rather than having something that is 70% complete and having a modest amount of contamination. So we'll accept this sliding scale. If you look at the spots, they cover a huge area, but in terms of the density of these spots, I'd ask you to look at the right-hand side where we actually have the counts according to that particular area. So what you'll see is down this bottom right-hand corner, we have nearly 40,000 high quality, and we have over 50,000 of this medium quality, this QS50, so the ones that have this trade-off between completeness and contamination. So for the next part of the talk, we're just going to focus on the analysis of these high quality. We then did much the same with the medium quality, but just for the sake of clarity in numbers, we'll focus on the high quality. So we've done the check-m approach, and we think they're high quality. Now, what we then did is start to look at complete isolate genomes to compare them. So to do this, we constructed 2 different databases. There's RefSeq, we just took RefSeq, but we also took all of Trevor's data sets of isolate genomes, plus those from HMP that were isolated from human gut, and any others that we could find that we knew were from isolated from the human gut. So this became the human-specific reference catalogue. Now what we could do is try and compare these isolate genomes to each one of these bins, but due to the sheer weight of numbers, we can't do an all against all alignment, so what should we just use MASH? We've heard a lot about min hashes. MASH is a tool that uses this sort of approach for finding a first approximation of the best genome that's going to match our bin. When we take the best one, we then go through and run DNA diff, part of the MUMMA package, to actually work out the fraction of the alignment and the reference that match one another, and then the average nucleotide identity of that alignment. And then this also allows us to infer taxonomy, so we can correlate these 2 tools. So if we take those high quality mags and look at the fraction of the mag against the reference, this is the plot we see for these ones that were all considered high quality according to check-m. Now if you look in the top right hand corner, that's where we considered we had a good MASH match. And so when we look at those, and if we consider a good MASH match to have more than 95% ANI and at least 60% alignment coverage of the query. And if we take those and then drill down into these a little bit more and plot the average nucleotide identity, i.e. those that are greater than 99%, so that should be strain level. similarity or identity. Then the light blue is the species level, and then grey is something beyond the species, so something more distantly related. So what you can see here is actually there's a lot of points that are actually where we've de novo reconstructed something that's equivalent to an isolate at the strain level. We have many more that are equivalent at the species level, and you're getting large fractions of alignment of the mag to the isolate. If you look at the diagonal, it's just slightly down on the axis, and that's what you'd expect because we know that not everything will perfectly assemble. Things like the ribosomal RNA typically doesn't assemble out when you're doing these. So this gave us a lot of confidence that when we get a good match, that we're finding a very good match to that isolate genome, and that CheckM was a pretty good approximation to complete for the quality of completeness and contamination of that genome. There was also this big cloud of grey points over here. Those grey points were the things that interested us most of all. So I showed you the results of using that human-specific gene catalogue. If we use RefSeq, we get similar sorts of results. And if we start looking at the intersect here, out of the near 40,000 high-quality matches, we find that in Trevor's set, we can match about 15,000 of those mags. 11,000 can be matched by RefSeq, showing that Trevor had a lot of novelty in his set. There's about 1,000 genomes in RefSeq where we get a good match, but they haven't been isolated from the human gut. So they're from another source, they're maybe from another body site. Some of those are oral, and we find them in the human gut. Some of them are just related and are found in other sources. But there was also nearly 30% were unclassified, that grey stream. If we still just focus on the classification of those things that we can match to an isolate genome, here, if we plot the phylogeny of all of those isolate genomes according to their 16S, and then just put the occurrence of matches to the species that we have for the mags, what we find is we see the same mag over and over again for many of these. So we have very good coverage of this phylogenetic tree, so that this colour code indicates a different class, and these whiskers outside indicate the frequency of that mag or that has matched that species. So here again, what we've been able to do by taking many, many different samples, we've independently reconstructed the same genome. So what we're getting is we're getting multiple observations and very good matches of these de novo assembled genomes against isolates, but there's still this set of nearly 12,000 that we haven't got a match for. So what are they? So as you've realised from that previous plot on that phylogenetic tree, we have a lot of redundancy within the system. Now we have to now remove that redundancy amongst the things that we can't match. So to do that, what we're trying to do here, I've got a little picture at the top of our bins, and so there's 3 green bins, and the colours representing something that's the same. We've got a red one and a blue one, and we want to remove the redundancy of that. So we use, again, MASH and DREP to do some clustering, and then choose the very best one to have a representative species out of this unclassified set. So in the case of the green one, typically what you'll be looking for is that one that has the most completeness of the genome. And so it uses CheckM again and chooses the very best one. It uses some other metrics, and it tries to minimize the contamination. So once we had that representative species, we could again look at the taxonomy assignment. But we could also do some more analysis. We could use protocol, and then we can start looking at the phylogeny of those. So we can take a set of marker genes to have spec I. We can't use 16S because it doesn't consistently assemble out, and we can start looking at the function of those. So to cut a long story short, we took that, what I've shown you is for the high quality mags, we took the low quality mags and added those in afterwards. So we took those high quality in preference and then dereplicated the low quality. And in total, we identified nearly 2,000 novel species, so 1,952 novel species. If we put that in context to the species level of Trevor's data set, so I said it was 739 genomes, there was lots and lots of strains within there. So if we actually just cluster that at the species level, that's 553. So that's over 3 times the number of genomes that we've managed to recover de novo that are novel, unclassified species compared to what we know at the isolate level. That was quite a lot. So, what could we find and what could we say about these? So, we're really cautious about not overstating things, and so we always tune what we say to the sort of more conservative side. We also do a lot of work to try and verify the classification. So we can use CheckM, that gives us an idea. We can use a lot of phylogeny, but we also inspect the bins and use things like CheckM, Diamond, and Uniprot to make sure that all the proteins are consistently annotated to the same level, to then verify the taxonomic placements that we use. So here we can take those 1,952 and infer actually that 60% of those we can actually place in a genus, 40% you can't. It depends if your glass is half full or half empty. We don't think there's any novel phyla. If you ask a typical person whether the phylum distribution of these novel species is what you'd expect in the gut, I've not yet found anyone who's disagreed with that sort of distribution of what you typically find. So we don't think there's any novel phyla. You see a little gap at the top, and that's just where either due to incompleteness or the distance or the sensitivity of the marker gene profiles that's used in CheckM doesn't perfectly identify enough marker genes to give reliable placement. So we think we know what they are. We can place them in the tree, but we just have some degree of caution. But we don't think there's any novel phyla. If we actually look at it, many of the species that we've identified come from this Collinsella genus. So that's this part here. And this is really interesting in the fact that Collinsella is actually a genus that's known to be important for gut health. So that immediately sort of made us quite interesting, but also slightly surprised that, you know, we've got a lot of healthy individuals, but why haven't we seen so many of these genomes before? If we put this in a sort of more global context of the human gut microbiome, what we know today, we can draw a phylogenetic tree using speci and the marker genes. So this is 40 marker genes for drawing this tree. Around the outside is, in these various colours, are the phylum. Inside you'll see these different colours coming off the tips of the tree. Now blue means it is an isolate genome. Green means that there is a high quality, so more than 90% complete and less than 5% contamination MAG, and red is that QS50 set. So what you can see is actually over this whole tree, there's very good interspersing of what we find as novel with the isolate genomes, with possibly the exception of here in the firmicutes, but that reflects where Trevor did a lot of targeting. So these are overrepresented in the isolate, so he spent a lot of time doing that. So we have equivalents in this region, but we're just showing you we've got an isolate genome there. Around the outside are a couple of interesting things. So what we've done is we've mapped the reads back onto this set to look for the prevalence of these, and we can also say whether a particular genome is confined to a geographical range. So the red indicates where it's non-European, non-North American, and blue is where that genome has been found across all geographic ranges, all 6 continents. So what you'll see is there are not so many that are ubiquitously found, but there are some. But there are also some genomes that we've pulled out where they seem to be very skewed in terms of geographical distribution. And the reason why we looked at non-European, non-North American is actually those samples are particularly underrepresented in what we actually could analyse, and we'll come back to that in a later slide. So there's lots of diversity in this, especially amongst the firmicutes. There's this slight strange region here, which is in the actinobacteria, so this column-cellar region. And you might think this is a bit strange. We think that actually there's something going on with the column-cellar group. We see this in the isolate world as well, and the fact that there seems to be a lot of, it's very difficult to get a good species delineation of these genomes, and so you get this slight strange part of the phylogenetic tree. We also had genomes for 4 phyla where we knew from 16S they were present, but we've never had an isolate genome from the human gut. So we've got now representative genomes for 4 novel phyla. So, what can we start saying about these from a functional perspective? So we've got lots and lots of genomes. So we've used a number of different tools to look at the functional profile of these. So one of the tools is something called Genome Properties. So this was actually developed first of all at JCVI in the US. So this uses TigerFAMs and PFAMs, and a little bit like SEED and KEGG, but has slightly broader application. It uses those protein families to infer the presence of pathways and systems within a genome. So what we can do is we've got here a PCA of the presence or absence of those pathways that we've found. And in this PCA plot, we color code according to phylum, but there are different shapes in there. I don't know if you can resolve that at the back. It's reasonably, so we have a square indicates an isolate genome, an X, a near complete genome, and a circle, a filled circle, these medium quality. So from this initial PCA plot, actually we were quite relieved, in fact, that there's no obvious separation between the isolate genomes and these MAGs. So even if you've got the lack of completeness, it's not separating out significantly. So that's not the biggest signal. The biggest signal is the function of the phylum, which is great. So it means you are recovering biology. But what we wanted to do is go a little bit further and a bit more fine grained. So when we start looking at genome properties at the different levels, at the phylum level, we do start seeing a separation in some cases. So here it's the same symbols, but just to make things a bit clearer, blue is isolate, red is near complete, and orange are these medium quality. And you can start to see patterns of separation, probably with the exception of the Bacteroidetes, where it's a bit more mixed up. So you could say, well, maybe genome properties is skewed. We've looked at the same thing for KEGG, these higher order. So here's the output of ghost koala. Generally, we see similar sorts of patterns, but if we look at just the number of enriched genes, there is definitely a distinction between the MAGs and the isolate genomes. And so we were like, okay, so there's something there. Can we go one level down, even more fine-grained? So what we did is we started looking at functional terms assigned to the proteins. So we used interpro for this, and then assigned Go terms from those interpro, and grouped these according to the top level Go terms. So to try and to identify any functional properties that were over or underrepresented in the MAGs compared to isolate genomes, we took those as 2 distinct sets, and then we can then use effect plots to try and identify things that are either overabundant or underabundant in the MAGs. And that's what you've got here for 4 of the different phyla. So actinobacteria, firmicutes, proteobacteria, and tenurocoutes. So if you've got a positive effect, you're more abundant in the MAG. If you've got a negative effect, you're less abundant in the MAG. Now hopefully you can see at the back, if we look across all 4 phyla, there is a common theme to some of those groups that are underabundant or less common in the MAGs. So these were all typically, if you look in bold, all of these, so cell redox homeostasis, oxidoreductase complex, antioxidant activity. These are all things that deal with oxygen and the ability to actually tolerate that. And if we actually think about it, what we think is actually these mags, one of the reasons why they're probably not being seen in the culture is that actually they seem to be very, very sensitive to oxygen. So when you have a stool sample, that's a proxy to what's going into your gut. It does get exposed to the air no matter what you do, pretty much. There's very few direct biopsies. They are going to get exposed. If these are very, very sensitive and you try and plate these out, they've either been damaged or killed by exposure to air, then you're not going to be able to culture them because they're just not going to grow. And if we, to just reinforce that as well, we see some of the things, so iron binding as well starts going up in iron sulfur clusters that can be used as different redox potentials. It becomes more common in these mags, reinforcing that observation. We also wanted to look at geographical distribution, as in we've already highlighted that some of the mags seem to be only found in non-European, non-North American populations. Some of them are found ubiquitously. So what we've done here is we wanted to, if we look on the left-hand side, these are color-coded according to the country. So green is North America, Europe is blue, Asia is this sort of red color, Oceania this sort of yellowy color, Latin America this purple color, and Africa pink. So what we did is, as we added more samples in, we asked how many new mags did we see as we added more samples in from these different continents. So North America is the most sampled geographically, Europe thereafter. So we have many more of those. So as we add more of those samples in, how many new mags do we see? So if we plot this cumulative number of mags detected, what we see is as we add more samples, once you get over about 2,000, we started to shoulder off. For Titus, who's an American, what you'll see is there's slightly fewer mags we've identified, suggesting your diversity in the US gut population may be slightly less than European. Not saying anything about diet, but we just say that it's slightly less. So what we think is actually using de novo assembly for North American and European guts, we think we might be beginning to plateau. These lines are going to go on forever. You're always going to find new species just because there will be things in your gut that just come from food, etc. We'd never expect it to be flat, but we've certainly got past this big shoulder. If we look at Asia, it seems to be following a similar trajectory to the European gut, which is again quite interesting, but it's sort of, we still lack samples, and Asia is really biased in the fact that it's mostly Chinese populations that we see. If we look at the other continents, they are so poorly represented, you can't make any inference whatsoever. We just know that we're going up very steeply, finding novel things as we add more samples in. If we look at the read mapping, and I'll come on to the details of these finer ones a little bit more, but if we just look at read mapping back to this catalogue of isolates and mags, one thing that makes a huge difference is because we've found some novel genomes from Latin America and Africa, the read mapping makes a huge difference. So they definitely have a very distinctive gut microbiota compared to North America and Europe. I think we can say that quite safely. And that's sort of what we sort of know, but having a few genomes makes a huge difference if you're just trying a read mapping approach. The take-home message of all of this work really was we've got a lot of, 2,000 novel uncultured bacterial species. We've got 40% that were novel genera. But and as I just mentioned, it improves the read classification. But if we actually look at read classification of everything, say all of those samples, if we just take the isolate genomes, you can map about 55% on average of all the reads to a genome. with a huge variance. So this is, the HMP project was a multi-million dollar project. Trevor spent a fortune isolating, but you're still only representing a small fraction, well, just over half of the reads could be mapped. If we add in the extra genomes we had from RefSeq that weren't isolated from the gut, we can push that up slightly more. But then as we add in the mags, the high quality and the low quality, that changes from 55% up to 75%. And you'll also notice the variance really starts reducing down. How much more do we think we could go in there? We know there are lots of things that aren't represented by bacteria, but again, this sort of is in agreement with this sort of shouldering of the plateauing of what we see for the sort of North American and European guts, because that's biased. You can sort of carry that forward to this analysis of the fact you can say so much more. So we think that is getting there. We don't think we've got everything because there is limits to the resolution of de novo assembly, but we think that we're getting a lot better. So we're not the only ones. So Mick kindly pointed out that Zagata's paper came out about 2 weeks before our paper came out, and then a couple of months afterwards was one from Nikos at JGI. Again, having done a similar thing, but used different methods. Zagata's used a very similar pipeline to ours. He must have nicked Mick's pipeline as well, because he keeps saying I've stolen his pipeline. And so the question is we're all about reproducibility. So we think we're doing okay. So we've managed to compare to isolate genomes and we're doing okay. We've seen multiple observations, but that was in our hands. If we compare to other groups doing this, what's the overlap? So before I go and look at that overlap, one of the things that we care about and the reviewers cared about and kept on us, and also in discussion with the other groups, had asked exactly the same question in review for those, is how reproducible, if you use a different pipeline, do you get the same answer out? So I think one of the most interesting things is the headline figure is yes, you do. So we've got two different things that we looked at. So one was to use a different assembler and actually perform co-assembly. Different assembler with different binners, that's a meta-wrap, and then to perform co-assembly. So this is where you use multiple samples to look to see if you can actually improve the mags. This co-assembly is a slight modification of standard co-assembly, but the principle is the same. So what we've done here is we took a subsample of everything, because we weren't going to do everything. We took 1,000 samples and looked to see did we get any improvement. So here what we have out of that 1,000, for the co-assembly, we had something like 4,000 mags that we wanted to compare to our set. And then we compared those and we said an excellent match was where you've got more than 98% ANI and more than 80% of the genome covered. A good match was 95 and more than 60%, and then no match was anything else. What you see here is using a different co-assembly approach, you don't actually gain much. There was a couple of genomes where you got something out of the co-assembly where we didn't have it. That's where it just pushed it over that QS threshold. You just got a couple more contigs and you just pushed it over the threshold. But generally, it wasn't making a huge difference. You actually saw slightly more when you used of a difference when we used meta-wrap, but we also used megahit as an assembler there. Now we were able to go in and dissect this. This is actually, the improved performance actually comes from using meta-wrap, which uses 3 different binners and then has a refining procedure for that binning approach. And that actually made more of a difference than actually the assembler in this particular case. This is obviously only looking in one direction. If you look in the other direction, the Metaspades people would be pleased to know that actually they outperform megahit by a reasonable bit. So take that. If you're wanting to do assembly, don't use megahit. Mick, didn't steal your pipeline. Okay, so if we take all of the different de novo assembly sets, all these mags from Sagata group, ours, and JGI. And then we applied a uniform metric. So everyone's got their slightly different way of having completeness and contamination. We applied our slightly more stringent completeness and contamination compared to the others. So this is having this QS of 50. And then applied it to all of the mags and all of the isolates. Now, the number of isolates has gone up here. So what we've done is, through this work, we've gone to all of the different genome databases we could find. So we've not only used RefSeq, we've gone to Patrick and tried to identify any match at the species level. So here we can get a few more genomes out. There is redundancy within all of these sets. And if we take all of these together, that's nearly 300,000 gut genomes. There's huge amounts of redundancy of that. We can apply the same dereplication process to everything, preferentially keeping the isolates, and that comes out at 4,644 species clusters. Now, that's a huge reduction from nearly 300,000 down to just under 5,000 at this particular level, and there's a lot of clustering. 81% of those does not have an isolate known for the species. So this gives Trevor his line in the sand of how much more work he's got to do. So he's done, just a mere 19%. The whole community's done 19%. Okay, so there's huge redundancy. So is everyone occupying a slightly different space? So we started to look at this, and here is some really early results off the doors. So here what we've got are the number of the species that have been carried through from, again, Sagata's group, my group, and JGI. And these are the numbers after all of this dereplication that have gone through. And what we've asked here is how many are found in all three sets, and how do they correlate with the isolate genomes. So the good thing is that the largest group is actually where across those species over 1,000 are found in all three sets. Next group after that is a unique set that's found in Sagata's group, and when we look at this, where these have come from are actually where they've used different samples. So there are a number of different samples where there were quite interesting geographically diverse regions and just some additional samples. If you just keep going through, there's essentially lots of overlap in the mags, and then interestingly again is that the isolate world seems to have found some unique areas where we're not picking them up in the mags. They're not found. And then there's lots of overlap going on thereafter. If we just look at the mags and then just the samples that are in common and the mags from those samples that are in common, what you'll see is that across the three groups we've got a huge overlap. So here the vast majority, 798, were found across all three. Now JGI used MegaHit, and that actually, so EBI and Sagata's group were both using metaspades, and that's the next group that overlaps. And this probably reflects a slight difference in the experimental approach, the informatics side, where we get slightly more there. Then there's a group, again, where JGI and Sagata have slightly more mags coming through. I think this is just due to our pruning off, but essentially you're seeing very little where they're unique in the overall data sets when you take all samples that are in common. So how good are these overall? So the beauty of when you do the dereplication is you take the best representative of everything. So in this particular graph, we've plotted the completeness and contamination, but we've just limited it to this 5% contamination. And actually, if we look at the overall completeness of everything, we have 95.7% completeness and less than 1% contamination. On average, there's 19 tRNAs, which is pretty good. What's not so good, but not surprising, is that the number of ribosomal RNAs, the 16S and 23S, is only found in about 1,000 of the 4,000. Now, within the MyMag standard, the very, very, very good quality is those where the genome has both the 16S, 23S, more than 15 tRNAs, and completeness and contamination greater than 95% complete and less than 5%. If we actually group these here, there are very few, and these are mostly isolate, although we do see some mags where you do recover the 16S and 23S, but they are very rare cases, and they are cases when, if you go back, they just seem to be very dominant in that sample and manage to just get through the assembler. But overall, what we find is if we look at the proportions, everything is where you want it to be. So we think we've got a pretty good comprehensive set of the human gut microbiome now that's usable for driving further analyses. We have a long and deep discussion about what you should and shouldn't do in terms of how far should you push this. So here is an example of what we're doing at dereplication. And in this particular figure, what you've got are the, I think it's 30-odd Sakari bacteria that we've managed to assemble uniquely. And if we look at the clustering for DREP, so it performs this hierarchical clustering, so you have an initial grouping. Here, you cut that off, and then you choose representatives at the 95% level. So in this grouping, this green grouping, you have one isolate chosen, one mag chosen, and in this top part, you have 6. If we actually align all of those, you see a lot of variation in the genomes. What that exactly means is still, we don't know. These could be quite important variations. There will also be common things where the assembler doesn't work, so things like 16S. But we want to capture all of this information, but also not overwhelm people because of just the sheer weight of numbers. But it is worth noting that actually there's a lot of variation amongst these genomes. To sort of try and encapsulate that, what we started doing is actually taking this combined set and actually looking at the pan-genomes of those. So if we take these 4,644 clusters, that actually, if we take all of the mags and all of the isolates and look at the novelty of those, that's 625 million proteins. Just to put that into perspective, Uniprot is about 150 million proteins. But there's huge amounts of redundancy in there. So we can then just cluster those at 100% identity, and that's 171 sequences. And then if we go at the 90% level, 14 million sequences, and 50% is 5 million sequences. It will come as no great surprise that the more genomes that you have, so this graph is looking at the number of core genes, so the more genomes that you have that represents that species, the fewer number of core genes. So it's inversely proportional to the number of genomes, the number of core genes. And that's not really surprising. As you add a new genome, the chances of that protein being found across everything, there are some, but at that sort of clustering level from Rory, so that these core genes diminishes as a fraction of the actual proteome, so you're just left with the core housekeeping genes. But if we look at the overall pan genome size, if we look at the number of genomes that have represented that species, and actually the total pan genome size, actually there's not so much that's correlated. And so some are massively expanded. Some of the species are massively expanded when you look at all the genomes, and that's not just purely weight of numbers. So what does this all mean, and how can we do this? So there's lots of different approaches you can take. I just illustrate with this one fairly topical one. So a scientist came out a couple of days ago, so 14th of June this year, so announcing that the FDA have ceased FMT trials because of the fact that if you drill down to the details, but essentially the fact is that when 2 patients got contracted antibiotic resistant infections from an FMT, and one of those subsequently died. I'm not exactly, it's a little bit hard to work out whether they died because of the infection or other factors, but essentially this illustrates the danger. So if you just blindly take the stool sample from a donor and put it in someone else, you don't know what you're necessarily transferring. So one of the things we can do is we can go to this collection and look at antimicrobial resistance across what we've discovered. And this is just looking at the species representatives. Here you've got the phylogenetic tree. The outside with green whiskers is a pan-genome size, and then the number of antimicrobial genes. So we use Abricate for identifying this. It will come as no surprise that beta-lactams was the most common antimicrobial resistance, but this just shows you that actually the spread of AMR across what we've identified is not uniform. There are distinct pockets that some of it seems to be correlated with pan-genome size, so you would probably say eyeballing over here, there's definitely a distinct correlation. Probably over here, so you should also note that this taxonomy is changed to the GTDB, hence the firmicutes is split, but in this firmicutes here, again, it looks pretty well correlated for the pan-genome size and the number of AMR genes. However, there are cases where it doesn't correlate at all. So in the proteobacteria, this doesn't seem to be the case. And this is just looking about the AMR that's encoded within the genome. So there are skewed distributions, which will be important for people who are wanting to think about therapies and wanting to take genomes and capture those and make drugs. However, the upside is that the human gut microbiome is well known to also be a rich source of novel antimicrobials. So there are lots of different antimicrobials. A lot of them are encoded in biosynthetic gene clusters. Here is a non-ribosomal protein that's actually an antimicrobial, and what you find are these cassettes where it's multiple different genes can carry out different functions to produce this particular manellomycin antimicrobial, which is a non-peptide non-ribosomal protein. So we look just in the EBI mags and Trevor's isolates and look for this. So we used anti-SMASH, which covers all biosynthetic gene clusters, and we look at the counts. The first one's not particularly interested, so anti-SMASH covers things like quorum sensing, but if we look at the next two, you've got a huge number of novel non-ribosomal protein synthesis and bacteriocins, both of which are potential. And so one of the key challenges is that how do we start working out what's truly novel or just variants of the things we've already seen? And the mags also seem to be slightly enriched in these compared to the others. I don't know how statistically correct that is. That just leaves me to sum up and just say thank you to my team, which is represented here, particularly Alex Almeida, who's done a lot of this work. I had many key collaborators in this work, particularly Trevor and Sam Forster, Greg Glor, and obviously the Sagata groups and Nikos's group of combining an access to E&A's wealth of data, of which is not a direct access MIC. And thank you very much for listening. I'll take any questions.
We have time for some questions.
Is there any trend for this non-ribosomal proteins to belong to taxa?
Yeah, there is a skew in taxonomic distribution of those.
What for?
I can't remember off the top of my head exactly which group. They do generally have a slight skew in distribution. Some of this is about just colonizing, grafting. A lot of the firmicutes have them, as I think is.
I have a question. One of your slides you mentioned the level of contamination is less than 5%. How do you define contamination in metagenomic data sets?
So this is using the single copy marker genes. So this is very much looking to see if you have a single copy marker gene more than once. And then that gives you an indication that, because you should only see it once.
So could you go to that slide? Is it
Which one?
the one with the contamination cutoff? You have that sort of green and blue box on the lower right.
Yeah, really early on.
Yeah. I think you can hit like 1G and it'll go to like the first slide.
This one.
Yeah. So as a result of our sort of graph query thing, I didn't mention this because I don't know how to defend it. When we do our neighborhood query, we in exchange for increasing the completeness of our bins, we also bring in a lot of the contamination.
Yep.
When we look at the contamination, the domains are all pretty close. So I'm sort of curious if you have any thoughts on this check-in,
Yeah, I have lots of thoughts, and I think I can share those thoughts because CheckM uses PFAM profiles underneath, so I can then criticize PFAM. So a lot of those single copy marker genes are really, the profile HMMs are really skewed because they're built off what we know in the reference databases. So first of all, often your completeness is skewed, especially when you're going into novel areas. So probably for the human gut, they're not probably so bad because you've got reasonable representation. But for more diverse environments like marine and soil, those profiles are not going to do so well. In terms of the composition of those, of what we consider a single copy marker gene, again, they are skewed because of the fact that it's what we know from in the reference database. So some of these are genuine gene duplications, and I don't necessarily think they're that bad, but we have to use something in the absence of. But I think what we showed, and that's why we were really keen to use the isolate genomes as a comparator and be reasonably confident. But I think that in the absence of any metric, CheckM is not bad. I think that there is opportunities for tool development in this space. There's a lot of KMA profiling, etc. We using things like the multiple binners, they get better with the refinum. I think there's an element there where that'll actually refine the bins. I'm sort of slightly waffling now, so I'll wrap up and say CheckM's not bad. It could be better. There are different approaches, but I'm not going to develop them for bacteria.
Hi Rob, I just have a quick 4 questions.
Great.
No, they are quick. Either you didn't find any archaea or you just didn't...
We have got archaea.
Okay.
We just didn't bother with them at the moment because so they're virtually all one group.
And very right.
Yeah, exactly. So they're virtually all clustered down to one species. There's about 4 different archaea. We're all in the same genus.
So one of the things we've been battling is where to draw the line of what represents a new genus. So one of the metrics I've come across in the literature is 60% amino acid identification AI. So have you kind of looked at that and whether that holds up? It doesn't hold up with the current taxonomy, but let's see.
Yeah, well, have you used the GTDB? Yeah, I think it holds up better.
Yeah.
We have not extensively looked at it. I mean, I guess where we went with the protein side is looking for the annotation across all the proteins to make sure they were consistent. I think we just relied on the ANI. I mean, yes, so the AAI is also a metric, but I don't have a good feel for the level of AAI.
Hopefully you will review our paper when it comes out in a few weeks. Remapping or reclassification? Because your graph said reclassification and your voice said remapping.
So, well, we use that sort of interchangeably in terms of, there's remapping, but we'll only classify if you've got a good level of coverage for that genome.
Yeah, because
you get a lot of spurious, you can have a lot of spurious matches.
So I guess in my head, reclassification is just you find one, like cracking classification. If you want to find one KMA in the read, then it's classified. But remapping is you're trying to align the whole read.
Yeah, so we're trying to align the whole paired end read, and we're also looking for a certain level of coverage.
Okay. I recommend you stick to mapping, then that's probably the thing.
Sorry.
in the nomenclature. And then, right, final question. There were 56 genomes that were found in all 8 or whatever the studies you've heard about. All of the culture ones and all of the manual ones. There were 56 that were found in every single one. Do you know what they are?
Not off the top of my head.
I mean, that is literally the core.
That is, yeah.
Of all bodies.
But so I would disagree with that statement because of the fact we have many representatives from each continent, and so it's the fact it's been found once, it's not the fact it was found in every single sample from that continent. So it's not quite the core, but they probably are fairly, the fact that they are ubiquitously found probably would be, they're not a bad starting point.
I'd be really interested to see what they are.
Okay, I think one more question.
Is there perhaps in development or existing a more automated way to merge mags? Checkman has a merge mags feature, but then it's a manual curate.
Well,
is there anything in the pipeline that would look at the biology perhaps?
I mean, there's two. The question is are you trying to refine them, i.e. remove contigs that potentially shouldn't be there, or are you trying to take two bins and join those together? So to the first part, the MetaWrap does this refinement process of throwing away contigs. In terms of merging to get a better coverage, there's nothing I know in terms of you can look at, other than looking through this manually, I don't think there's anything that's really automated, because there are various tools that can give you hints, but nothing that's purely automated. This becomes more important for eukaryotic. So eukaryotic genomes, I've not talked about. We see them across lots of different microbiomes, less so in the human gut due to extraction methods, but they're merging across bins, or merging bins to cover, is important.
Okay, let's thanks again.